DNA cloning. Procedure in which a fragment of DNA is inserted into a vector and then transferred to another cell, where it then replicates. Multiple copies of a single gene equals gene cloning. Restriction enzymes make recombinant. Cut DNA at specific nucleotide sequences. Instead of cutting the molecules straight across, they leave sticky ends. Any DNA cleaved by such enzyme can be joined readily to another DNA molecule cleaved by the same enzyme. **** DNA ligase is used for permanent union. Plasmids and circular, self-replicating, non-chromosomal DNA molecule found in many bacteria, capable of transfer between bacterial cells of the same species, and occasionally of different species. Antibiotic resistance genes are frequently located on plasmids. Plasmids are particularly important as vectors for genetic engineering. Bacterial artificial plasmid. A plasmid in bacteria that can hold large segments of DNA, greater than 100 kilobits, and contains genes for partitioning the plasmid to daughter cells, antibiotic resistance genes, and an origin of gene cloning in backed plasmid. Transfection. Electroporation, a technique to introduce recombinant DNA into cells by applying a brief electrical pulse to a solution containing cells. The electricity creates temporary holes in the cell's plasma membranes, through which DNA can enter. Viruses, gene guns, the needles, can also be used to transform cells via injection of DNAChemical methods can also interact with the cell membrane to make it permeable to DNA insertion. cDNA from mRNA procedure. Fully processed mRNA is extracted from cells where the gene is expressed. Reverse transcriptase, obtained from retroviruses, is used to make a single-stranded DNA reverse transcript of each mRNA molecule. DNA polymerase is used to create the complementary strand. A tail allows use of short strand of DTs to be used as primer, equals complementary DNA, cDNA. cDNA library versus genomic library. cDNA libraries won't include introns, exons or other regulatory elements, but a cDNA library is useful for studying sets of genes expressed in particular cell types, also making cDNA at different time points in a cell allows tracking of patterns of gene expression during development. Nucleic acid hybridization. A technique in which a single-stranded nucleic acid probe is made that is complementary to, and binds to, a target sequence, either DNA or RNA. The resulting double-stranded molecule is a hybrid. The probe can be labeled with a radioactive isotope, a fluorescent tag, or another molecule for tracking. Polymerase chain reaction (PCR). DNA amplification in vitro (PCR). Double-stranded DNA containing target sequence heat-resistant DNA polymerase, all 4 nucleotides, 2 DNA primers for target sequence, 1 for each end, equals greater than. Thermocycler, 1 cycle yields 2 molecules, 2 n where n is cycles, 1, denature, heat denatures DNA. Separate strands, 2, anneal, cool allowing primers to form bonds with DNA. 3, extension, DNA. Polymerase adds nucleotides to 3 end of primers extending sequence. Gel electrophoresis. The separation of nucleic acids or proteins, on the basis of their size and electrical charge, by measuring their rate of movement through an electrical field in a gel, such as agar. DNA moves towards positive pole due to their negative charge, phosphate groups. Southern blotting. Gel electrophoresis. Nucleic acid hybridization equals detect bands that include only parts of gene of interest. 1. Prepare fragments. Restriction enzymes digest samples. 2. Gel electrophoresis. Pattern of bands created. 3. DNA transfer. Blotting. DNA is transferred to a membrane creating a blot with a pattern of DNA bands just like those of the gel. 4. Hybridization. The blot is exposed to a solution containing labeled probe. 5. Detection. Sheet of film is used to detect hybrids. Western blotting. Technique that uses antibodies to detect the presence of specific proteins separated by electrophoresis. Gels versus gels. Agarose gels, DNA, RNA electrophoresis, uses size, compare to size ladder, visualize with ethidium. Bromide, polyacrylamide gels, sad face, SDS, paged proteins, uses size, blobbiness, compare to size ladder. Visualize with Kamase blue or antibody to Western blot. DNA sequencing is a technique for determining the precise order of the nucleotides in a DNA fragment. Dideoxy chain termination, Sanger method. Used to determine DNA sequence by inducing replication with altered bases that cause fragments of various sizes to be produced, creates a timed result printout that is read from bottom to top. bottom being the last nucleotide of shortest labeled strand. See image. Northern blotting. Steady expression of single genes, a technique that enables specific nucleotide sequences to be detected in a sample of mRNA. It involves gel electrophoresis of RNA molecules and their transfer to a membrane. Blotting, followed by nucleic acid hybridization with a labeled probe. Reverse transcriptase (PCR). Quicker and more sensitive than northern blotting, requires less mRNA. Reverse transcriptase yields cDNA, which is used as a template for PCR amplification using primers. Then, product is run on a gel, and copies of the amplified region will be observed as bands only in samples that contain the mRNA of the gene interest to measure gene expression. In situ hybridization, a method for detecting particular mRNA transcripts in tissue sections by providing a nucleotide probe that is complementary to, and will therefore hybridize with, the transcript of interest. DNA microarray assay. Organismal cloning. The same as reproductive cloning, a method to create a clone of an entire multicellular organism. This is done by taking a donor cell and fusing it with an egg cell whose nucleus has been removed. The fused cell begins dividing normally. This embryo is then placed in the foster mother's uterus and the embryo develops normally. Cell potency. Stem cells, and differentiated cells that continue to divide, they are pluripotent. Pluripotency. Ability to differentiate into many different cell types. Totipotency. Ability to differentiate into any cell type. Nuclear transplantation. A technique in which the nucleus of one cell is placed into another cell that already has a nucleus, or in which the nucleus has been previously destroyed and nucleated, can be used for reproductive cloning of a mammal, e.g. dolly. Cell reprogramming. Increase in potency. Dedifferentiation can be induced by nuclear transfer, cell fusion, genetic manipulation. Differential success rates in use of embryonic cells versus adult mammalian cells in reproductive cloning of a mammal indicates nuclear changes to cells over time with development. Epigenetic changes, acetylation of histones, methylation of DNA, etc., cause low success rates for normal development. Induced pluripotent stem cells, iPS cells. Any cell, even a highly differentiated cell in the adult body, that has been genetically reprogrammed to mimic the pluripotent behavior of embryonic stem cells, done via delivery of certain transcription factors, can differentiate into all three germ layers, ectomesia and end-derms. Telomerase activity. Stem cell marker. Still impossible to completely reverse all epigenetic transgenics. The study and practice of genetic modification by inserting genes from one species into the genetic material of another species. This can produce a new, designed species, can be used for protein production by farm animals. Human gene therapy. An experimental technique that uses genes to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery.